Thank you. I'm Lisa Forman from the University of Colorado, and I'm really happy to moderate this last official session of the day, which is about clinical trials and hearing from our pharma collaborators. As we were writing the guidance for PSC in the ASLD, which came out, I think, last year, the year before, we definitely emphasized that we thought it was essential for patients to be able to enroll in a clinical trial for us to really be able to kind of push forward and hopefully find a cure for PSC. And so really appreciate you coming to this conference and thinking about clinical trials. So and my first speaker is Dr. Joanne Quan from Mirum. She joined Mirum recently in 2024 as the CMO with responsibility for clinical development, clinical operations, and safety. She has nearly 3 decades of experience developing novel therapeutics, including those for rare diseases. And prior to Mirum, she served as Chief Medical Officer at Nuvig Therapeutics. And then from 2018 to 2021, she served in the same role at Modus Therapeutics, a company that was focused on rare genetic diseases. And before then, she had held clinical development leadership positions at Eiger Biopharmaceuticals, Intermune, Arena Pharmaceuticals, Bay Hill Therapeutics, Alza Corporation, Genentech, and Pathogenesis. So it's my pleasure to introduce her to talk about the studies that Mirum is doing. Thank you.
Thanks, Lisa. Appreciate that. Great to be here, and thanks to everyone for joining, and thank you to the PSC partners for inviting me to share this study with you. So I believe we have some slides. Hopefully we can pull that up. Oh, and where would that be? This one? Go. So here's the disclaimer, which you will read very quickly, obviously. Okay. Oh, let's see. Okay. First, just a word about our company, Mirum Pharmaceuticals. We are a little bit over 300 people. We're based in the San Francisco Bay Area. Our headquarters is in Foster City. We also have substantial operations within Europe. And we have a lot of expertise in liver disease, particularly in cholestatic liver disease. We have an approved drug, Maralixibat, or Livmarli, that's approved for a couple of rare pediatric cholestatic conditions, Alagille syndrome and what's called PFIC. So this is an area that we know well. Our mission really is to transform lives of patient communities. We're very much thinking about the voice of the patient, and so to come to a conference like this is perfect for us to understand what issues we want to be helping patients with the drugs that we're developing. So our drug is called Volixibat. It's what we call an IBAT inhibitor. And IBAT stands for ileal bile acid transporter. Within the body, bile acids are secreted into the gut and then are recycled. Most of it is recycled. And we know that with cholestatic conditions like PSC, the serum bile acids are elevated, and that can contribute to pruritus or itching. What Volixibat and IBAT inhibitors do is to stop that recycling and allow it to get rid of the bile acid within the stool. And so therefore, the serum bile acids come down, and our hypothesis is that this will actually decrease the itching in patients with PSC-related itch. So one of the interesting things about Volixibat is that it is pretty much stays in the gut, so less than 1% is actually absorbed. So essentially, it is taking the bile acid out of your system without actually pretty much getting to your bloodstream. So our study is a phase 2 study. It's called VISTAS. It is a global study. We have many sites in the US. You'll see that in a moment. This drug, Volixibat, has been tested previously in trials and has been used by hundreds of patients. In fact, it's been more than 400 patients have been exposed. We have some really nice kind of early study results. What we did earlier this year was that we had an independent data monitoring committee look at the drug, look at the safety, look at the efficacy themselves. We remain blinded to the results, so we don't know what those answers were. But we provided them with some guideposts. So we wanted to make sure that it was safe, that no people were being harmed by it, and also that it had a certain amount of efficacy. And they told us that it met both of those bars. And so we were kind of continuing enrolling the study. We also had 2 active doses and a placebo in this study, and they chose an appropriate dose, so we have a single dose that's active and placebo moving forward in the study. So we're very encouraged by this, and we're really excited to be continuing to enroll the study, and we'll hopefully be able to talk to a number of you about what the study is. Part of the study is after the 28 weeks of the randomized double-blind controlled portion, we have a long-term extension, and this is what we call an open-label extension, so that we know everyone who's participated up to that point will get an active study drug. So we think that will be very helpful for patients and also to give us additional data with long-term treatment. These are the basic study eligibility criteria. Obviously, we want patients who have PSC and also who are affected by PSC-related itch. We're allowed in the US to treat patients as young as 12. And so we do want to have some patients who are in the adolescent age group, and that can help us understand how this can help those patients as well. We have some exclusions, and we also have a booth outside. If you want to talk to anyone within the company about this, we can give you more information. This is a kind of busy slide of all the sites we have, kind of distributed throughout the US. One of the ones that I want to call your attention to is what we call the virtual site, and that's listed up on the top left there. The virtual site means that the site comes to you. You don't have to go to the site. We're offering many different options to make this accessible to people. You can either go to one of these sites here, which involves going to a study center, getting the evaluations, or you can say, I'd like to participate with a virtual site, and the site will come to you. We have home health nurses, people who can draw blood. Drug would get sent to your home if that's what's the most convenient for you. Or you can do what we call a hybrid option, have some visits at a study center and have some visits where the site comes to you. So we're hoping that this will make it a lot more accessible for people. We recognize that it is difficult to participate in trials, even for those who want to do so. So we're hoping that this will be an helpful option. This is just a little bit more in terms of the virtual site. It can be, the study activities can be done in your home or wherever it's most convenient for you, workplace, or wherever. When we do this, we establish a relationship between the study doctor in Vistas and your hepatologist or your gastroenterologist who's following you, so that there can be a sharing of information so that no information is lost. And we have a website there. We also have some folks at the booth who can help walk you through some of the study information to see if you might qualify. So just a couple last things. What we've done over the course of time is that we've tried to simplify the study screening requirements to make it a lot more accessible for patients. We have a great set of investigators who are working with us, and obviously they have a great interest in terms of caring for and developing drugs for PSC patients. We do cover travel costs. We have a long-term extension to ensure that any patient who wants to continue in the study and know that they're receiving drug will do so, and then also the virtual study visits. So thank you very much for your attention. Please visit us at the booth. Be happy to tell you more. Thank you.
And I think on PSC Partners' website, they have a list of all ongoing trials, and that's kind of another way for you to look and get connected to different centers that are offering these trials as well. So anyway, thank you. Next speaker is Dr. Praneeta Nagraj from Ipsen. Praneeta is a clinical scientist at Ipsen who specializes in clinical and medical development for rare diseases, and she's currently focused on phase 3 studies in rare cholestatic liver diseases. She's a trained clinical pharmacist who has spent the last 9 years in the pharmaceutical industry, and she has dedicated the past 4 years to rare cholestatic liver diseases, working in both the US and internationally in clinical development and medical affairs.
Thank you. Hopefully I can get this to work. So good afternoon. First of all, it is an honor and a privilege to be here. Thank you so much for having me. Today I'm excited to share a little bit more about who Ipsen is and a little bit more about what we're doing in the PSC space. This includes two current phase 2 trials and two separate therapeutics that we're under investigation right now. But before I begin, I would like to pause and say a big thank you on behalf of all of us on the research team at Ipsen, some of them pictured here. It is because of you that we do the work that we do, and we thank you so much for continually inspiring us, for people who are living with rare disease or caring for someone with a rare disease. So who is Ipsen? Ipsen is a global biopharmaceutical company that is focused on transforming and creating better lives for patients and people living with all kinds of conditions. We primarily have 3 therapeutic areas that we focus on: rare diseases, neuroscience, and oncology. In the last few years, we've increasingly strengthened our footprint within rare liver diseases specifically, and we have actually 5 focus areas of diseases that we focus on within that, and that is PFIC, or progressive familial intrahepatic cholestasis, Alagille syndrome, biliary atresia, PBC, and PSC. Of course, today I'll be focusing on PSC, and we're looking forward to what we have to share today. Why is our work important in PSC? I think I would be in good company to say that it is extremely important to do research in PSC. There are no currently available therapeutics. Our focus in our research is really focused on more of understanding potential mechanisms and therapeutics to address aspects of PSC, understanding what we can do to better liver outcomes and also health outcomes. This is my disclaimer. The purpose of this presentation is to give an overview of Ipsen and our clinical trials for PSC. The products here are investigational. Efficacy and safety have not been established and are also not approved by regulatory authorities. So our 2 therapies that are in development for PSC, it's a very small and busy slide, but I'll try and get through it and tell you how these work. So essentially on the left side of the graph, we have Ritavixabat, which is a novel, potential first-in-class oral systemic ASBT inhibitor. This is believed to work by inhibiting transport of bile acid in 3 places: the kidney, the bile duct, and the small intestine, or the ileum. By doing so, essentially reducing circulation of bile acids and accumulation of the bile acid buildup, which leads to liver injury, liver inflammation, and fibrosis. On the right side, we have elafibranor, and this is a PPAR alpha and delta agonist. The activation of this is believed to decrease bile acid synthesis and concentration of this. By doing so, again, reducing liver injury, inflammation, and fibrosis. So the first study I want to talk about today is Ritavixabat, which is in a phase 2 study. You see the clinical trial number on the screen here. This is currently recruiting as a study, but I do want to caveat that it's recruiting in Europe. If you'd like to take a picture of the slide, please do. This is a 12-week study evaluating the safety and tolerability of Ritavixabat with repeat doses once or twice a day in adults with PSC with or without clinically relevant bile duct strictures. It's an ongoing study recruiting up to 24 patients, and again, specifically in Europe. But I kind of want to go to the schematic to talk about what this study design looks like. This is a 4-arm open label study, meaning that all patients will be receiving active treatment with drug. So if we go through that schematic, upon screening and after enrollment, after screening period, patients will receive a single dose of Ritavixabat, after which there is a 2-week safety follow-up period where we'll be looking at PK parameters, after which all patients will go into the 12-week open label period with Ritavixabat across either of the 2 doses and into one of the 4 arms of the study. Then there will be, afterwards, a safety follow-up period of 2 weeks. This study is also looking at some exploratory endpoints, as it is a phase 2 trial. So some of those exploratory endpoints are things such as looking at FibroScan, PRO-C3, as well as ELF score. We have a second study in phase 2. This study is elafibranor, is called Elmwood, and it's a study with a compound called elafibranor. The study is a 12-week, double-blind, placebo-controlled study looking at the 2 doses of elafibranor, either the 80 milligram or 120 versus placebo, and evaluating the safety and tolerability in adult patients with PSC. I do want to say that this study was designed with the patients, with patient input and collaboration, and the reason for this is because it's incredibly important for patients to be involved in clinical research. So you have a voice in that, and this is one of the highlights of this program. When we look at the study design schematic, there is a 12-week double-blind period where you do have the 2 doses of elafibranor versus placebo, after which patients have an option to go into the open-label extension period, which is 96 weeks, followed by a safety period. That 4-week follow-up safety period could be at the end of their last dose or at the end of study. What's important here is this study recruiting. This study is not currently recruiting, but it is active. I am pleased to say that the Elmwood study is set for a completion date of the 12-week double-blind phase at the end of this year in December 2024. Similar to our other program, the study is also evaluating some exploratory endpoints looking at markers of disease activity in PSC, which we hope to give us some good signals about what's happening. So I know I may have rushed through the presentation, but I want to pause here and say I come to the conclusion of my presentation and want to say a big thank you on behalf of all of us. You are the reason behind the work that we do, and we're incredibly inspired by you all. Thank you for the opportunity to PSC partners. This is very helpful that we get to share some updates with the patients, and if you have any further questions about the trials, about the studies or the medications, please don't hesitate to stop by our booth and say hello. Thank you.
Okay. Next is Dr. Matt Frankel, who is the Chief Medical Officer at Chemomab. He is an internist and the Chief Medical Officer at Chemomab, where he leads the clinical development program of CM101, a first-in-class monoclonal antibody that showed promising activity in the recently reported Phase 2 SPRING trial in patients with PSC. Previously, he was Vice President, Clinical Development and Medical Affairs, Specialty Pharma at Boehringer Ingelheim, overseeing oncology, immunology, pulmonary, and CNS diseases. And at Novartis, he was Vice President and Head of Immunology and Dermatology Medical Unit, where he oversaw medical affairs and clinical development for several marketed drugs. At Sandoz, Dr. Frankel led the medical affairs unit supporting the biosimilar, biopharmaceutical, and generics businesses. And earlier, he held clinical development leadership roles across geographies and therapeutic areas in Rita, Fibrogen, Abbott Labs, and Schering-Plough. He received a BA from Vassar College and an MD from University of California in Los Angeles and an MBA from the Kellogg Graduate School at Northwestern.
Thank you, and good afternoon. I think you heard more about my life than my children know, so I apologize. I did not, but hopefully I'll educate them over the coming years. So you heard way too much about me, but let me talk a little bit about Chemomab and our aspirations. Chemomab was founded in 2011 by the current CEO and Chief Scientific Officer, Adi Moore. She is a PhD in molecular biology, and her husband, and they identified a chemokine, CCL24, which there's going to be a test later, so I'm going to ask you questions about these things. CCL24, and it was noted to specifically induce inflammation and fibrosis. And if any of you in the audience, I would expect most of you know that in PSC, both of those seem to be quite impactful on the disease. So over time, it was identified through a number of early assessments, whether it was in the lab and then ultimately in animals, that there really was an opportunity. So I'm going to walk through some of the situations. But CM101, I'll talk about the overview. I'm going to talk about the phase 2 program that we are excited to be able to present a little bit now, which is going to subsequently be presented at the American Academy for the Study of Liver Diseases in November. And then I'll talk about the phase 3 ever so briefly. And I'm going to do this in the next 3 minutes. Let me talk about CM101 and CCL24. As you can see here, CCL24 binds to a receptor on a number of cells that actually activates both the inflammatory cascade and brings in a lot of the inflammatory molecules that are essential for that. And at the same time, drives some of the actual fibrosis and the development of that. So if we can inhibit that, that's a good thing for a number of diseases, and specifically primary sclerosing cholangitis. And the CM101 binds that chemokine that binds ultimately to CCR3, and if you bind that, it can't actually fit with the CCR3, and then you don't get the downstream effects of the inflammation and the fibrosis. That was a lot. I promise there will be a test. So, this is the study that we recently finished the 15-week portion. So on the left-hand side of that, we had planned on enrolling approximately 70 patients. We ended up enrolling a few more than that. There were 3 arms, the placebo, a low dose at 10 milligrams per kilogram, and a high dose at 20 milligrams per kilogram. There was the 15 weeks, which was the double-blind portion, and then we transitioned everybody over to the open-label portion as well. Patients who could be enrolled were those who had large duct primary sclerosing cholangitis. They had to have an alkaline phos greater than 1.5 times the upper limit of normal. They had to have stable IBD if they had it. And if they were on UDCA, it had to be stable as well, which was the majority of the patients. We were looking at safety and tolerability. I'm going to give you a hint. It was considered safe and tolerable. And then I'm also going to talk about a number of the secondary endpoints. We looked at the activity in a number of ways. Now, I should mention again, this was a safety and tolerability study, smaller program. So the idea of being able to show anything of clinical consequence or clinically significant is very, very unlikely. And I think what I get to show today is some of the data that we found very exciting. There were a number of analyses. We were clearly going to look at the different dose and the dose response. We also looked at those patients who were more moderate and severe, so those patients who had an examination based on predominantly FibroScan where it was greater than 8.7 kilopascals. So those were in the moderate to severe patient population. And you can see where the study was completed. Let me see if this works. So I think I mentioned this. We achieved our primary safety endpoint. Secondary exploratory endpoints were quite promising, whether it's anti-inflammatory, anti-fibrotic, and frankly, the anti-cholestatic aspects, which you'll see. We achieved statistical significance in 2 aspects, and we identified a dose as well as a patient population that we think in patients that you probably know, and if you're not one yourself, who may qualify as well. The liver stiffness, which is the FibroScan, you can see that on the left is the all patients, and then on the right is about half of them who were the more moderate and severe patients. And you can see that the kilopascal went up in the, on average in the patients who were treated with placebo over 15 weeks, and it went down in the active arms. That's a good thing. And in the moderate to severe, there was even a greater difference between the two. So we really found that that makes the most sense for us to target those patients who are in that moderate to severe patient population. A striking feature that we found was the pruritus or itch. This was not a program where we specifically enrolled patients who were suffering from itch. In fact, most of the patients were in the mild to moderate space, so we were ecstatic when we saw that there was, in fact, a difference between those who were on the active arm, the lighter blue and the darker blue, versus the placebo, where there was not much change at all over the 15 weeks. Really exciting for us and hopefully for you as well. Another analysis that we completed was looking at bilirubin. So you can see that this is not comparing us, which you might think, to the baseline of the placebo. This is actually highlighting that in the placebo arm, there was no change, more or less, in the bilirubin. And yet, in those patients who received either the 10 milligrams per kilogram or the 20, that they showed a reduction in their bilirubin. This is encouraging as well. And then looking across all the labs associated with liver function, in the 20 milligram per kilogram dose group at the higher, meaning more moderate and advanced, you can really see a consistent impact across all the different liver function tests. Also really exciting. Going to touch on briefly something that you heard briefly, which was the ELF score. This is a composite lab test. It's been highlighted previously as a possibility of highlighting long-term outcomes. And I can say here again, we showed some encouraging data here. And then lastly, on the labs, there are two markers associated with inflammation and fibrosis, IL-6 and TGF-beta, and you can see a dose-dependent response. So across the board, really, really exciting from so many different ways. So you heard me mention these. I'm just going to highlight the last one, which is this gave us really an encouragement to move forward, and we feel confident in as far as the dose as well as which kinds of patients really would benefit most from this program. And based on that, we are meeting with the FDA. We're going to clarify our plans, and then we expect to have the phase 3 starting at the end of next year. We are also in the process of, you saw a number of the patients have most of the patients continued on the open label, and we'll have that data for early next year as well. So I want to say thank you to all of you. Thank you to the PSC partners. It has been a partnership, and we continue to collaborate, and we look forward to doing that in the future. And with that, I want to say thank you.
So last but not least, we have Dr. Richard Pencek. He leads the clinical development of the PSC program at Pliant Therapeutics. He obtained his PhD in liver physiology from Vanderbilt University and has 17 years of experience in clinical development, predominantly in metabolism and chronic liver diseases. He also was previously the lead clinical scientist for Intercept's cholestasis program between 2013 and 2019, which included studies both in PBC and PSC.
Thank you, Dr. Forman. It's a pleasure to be here with you all today. It's especially a pleasure because last year at this meeting, I was presenting on our ongoing Integris PSC Phase 2 trial. And today I'll be talking about our completed Integris Phase 2 PSC trial. Just in a moment of disclaimers, bexotegrast, the compound I'll be talking about today, is not approved for any indication, including PSC, and I am obviously an employee of Pliant Therapeutics. So in terms of bexotegrast and what its potential application in PSC could be, I think most of you in the room are familiar that PSC is a disease of the bile ducts, and it's this kind of static nature of bile in those ducts that leads to a persistent inflammatory response and damage, which can radiate out and affect the liver. Part of the signal in the damaged bile ducts is transforming growth factor beta. And under normal circumstances, this has an important role in repairing cellular damage in the liver and other organs. However, the persistent damage that occurs in PSC leads to a dysregulation of this component. And bexotegrast, and in some of these slides you'll see it referred to as PLN 74809, I think Bexotegrast has its issues, but is far catchier than that. It may prevent damage in these cells in the bile ducts and prevent them, essentially pump the brakes on this signal a bit to get it more under control and hopefully inhibit the progression of fibrosis in the liver of PSC patients. So Pliant Therapeutics is a small biotech company based out of South San Francisco. We are currently evaluating our lead compound, Bexotegrast, for the treatment of 2 rare diseases, one of which is PSC, which I'll be showing data for today, and the other indication is idiopathic pulmonary fibrosis, another rare disease, this one based in the lung. And that program is slightly ahead. The disease is a bit more prevalent than PSC, so it's a bit easier to find patients for an IPF trial in comparison relatively to PSC. Looking at the Integris PSC study, and this was our first go at it in PSC, so the primary endpoint was really built around safety and tolerability. We also had secondary endpoints on pharmacokinetics. This is really about looking at drug concentrations, how it's absorbed, how it's metabolized by the body. And then finally, exploratory endpoints looking at markers of liver health, fibrosis, as well as patient-reported outcomes and quality of life. The study as a whole consisted of a placebo-controlled evaluation of 4 different doses ranging from 40 to 320 milligrams, the bulk of which occurred over a 12-week treatment period. What I'll be presenting to you today is specifically data related to the longer-term treatment, which went out to a minimum of 24 weeks or up to 40 weeks for our highest dose level of 320 milligrams and the assorted placebo patients. To quickly recap the data that we saw through 12 weeks, Bexotegrast was well tolerated in participants with PSC. We didn't identify a concerning safety signal. Rates of adverse events tended to be comparable across all doses in placebo, if not slightly lower. And very importantly, there were no treatment-related serious adverse events with Bexotegrast. Based on the exploratory markers, it displayed promising signals of antifibrotic activity in these PSC patients with suspected fibrosis, as well as some additional findings which may or may not have been expected in terms of effects on itch relative to placebo with our higher doses. And we also had a note where alkaline phosphatase, something I think most of you will be familiar with, remained relatively stable over 12 weeks in comparison to a jump up in the placebo group. So the primary endpoint, looking at safety and tolerability, and this is specifically looking at the time period past 12 weeks with extended duration treatment with either Bexo or placebo. What we saw was that the overall rate of reported adverse events was comparable between Bexo and placebo, and also the rate of serious adverse events was low, with 3.7% of Bexotegrast patients experiencing a serious adverse event in comparison to 11% of the placebo. In both cases, these were serious adverse events of ascending cholangitis. Similarly, the severe events were the exact same events, and you can see a similar pattern with interruption of study drug. The most frequent adverse events in the study, we did the study during COVID. The most frequent adverse event during the study was COVID. And this was true for both groups, along with nasopharyngitis. Diarrhea and ulcerative colitis flares did occur at higher frequency with Bexo in comparison to placebo. These events tended to be mild or moderate in severity and usually lasted less than a day or a couple of days in general. Fatigue and pruritus, 2 symptoms which I'm sure many of you are very familiar with, were reported at lower rates with Bexotegrast in comparison to placebo. And cholangitis in general, while it didn't make the cut as a frequent adverse event, I think is very noteworthy that it did occur at 3.7% of Bexotegrast treated participants in comparison to 11% of our placebo subjects. Turning to some of the biomarkers in the study, first we'll look at alkaline phosphatase. This was a relatively provocative finding we had from the 12-week data. And to orient you to what you're seeing here, on the left graph is all the participants in the study. And there's Bexotegrast doses are shown in blue at 12 and 24 weeks, and placebo is shown in the gray bars also at 12 and 24 weeks. What we saw At 12 weeks was essentially no change from baseline with 320, but then by 24 weeks, we'd had about a 25-point drop in the alkaline phosphatase levels. This was in comparison to increases of 50 and 34 points that we saw at week 12 and 24 with placebo. And this finding was magnified when we specifically looked at patients who had an alkaline phosphatase above the upper limit of normal at baseline. Looking at the ELF score, ELF, if you've participated in a clinical trial, you may or may not be familiar with ELF, but I would say odds are really good you've definitely had it run. ELF is a score that's made up of 3 different biomarkers, and when you total 3 different biomarkers that are associated with fibrosis, and when you total these up to generate an ELF score, this has shown significant prognostic value with determining low, intermediate, or high risk of a PSC patient having a clinical event or progression of their disease. What we see for the population as a whole on the right graph is that this is relatively stable across placebo as well as the 320 milligram cohort. And we have an inclination in patients at higher risk of progression, so those who have a baseline ELF score of greater than 9.8, of a slight dip towards a negative value at week 24, albeit in a relatively small number of patients. MR, this was a voluntary sub-study that we included as an exploratory endpoint in the trial, and I'm sure many of the PSC folks in the room have had an MRCP at some point. This is basically the same procedure, but what we're doing with this is to look specifically at liver health and bile flow using MR technology. And essentially what's being done is that you're in the MR tube, you're going to get a delivery of your contrast agent, and the first thing we're going to look at is relative enhancement. So how well does the liver take up and hold that contrast agent? This is a measure called relative enhancement. Healthier liver is going to light up more essentially on the scan. And what you can see here is that from 12 to 24, we had an increase in relative enhancement. So things trending in the right direction with uptake of the contrast dye. Similarly, the other thing you can look at once you deliver the contrast agent is how long it takes to go from the circulation into the liver and then make its way to appear in the common bile duct. And we use this kind of as a marker of biliary flow. And what we saw over the course of we saw a tendency towards a reduction at week 12 that was further magnified at the week 24 time point, about a 100-second drop. For baseline information, patients tend to have a baseline of around 500 seconds, representing about a 20% drop. I think importantly with this graph, you'll notice that there are no gray bars. That's mainly because this was a sub-study. This was a voluntary, not every site that we had the capability to participate in the MR program. And the way this just happened to shake out with the patients who did participate, the vast majority of them were on bexotegrast treatment. We didn't know that because the study was blinded, and we didn't have enough placebo subjects to represent on the graph here. Looking at the itch, we looked at itch based on the NRS score. So what this is really looking at is literally asking patients about the severity of their experience with itch based on a scale, like literally asking them to score it on a scale of 0 to 10. And what we saw was essentially no change from baseline with Bexo at 12 or 24 weeks versus about a 1-point increase from 12 that persisted to 24 weeks. I will say itch in general was fairly low in the study at baseline, but still this is a provocative finding. So overall, continuing on from the original 12-week data, the 320 milligram dose was well tolerated with longer term dosing. Again, we did not pick up, we had a favorable safety tolerability profile, no treatment-related severe or serious adverse events with Bexo, continuing demonstrations of antifibrotic activity, as well as improved markers and symptoms of cholestasis around alkaline phosphatase, itch, some of our MR parameters, but in addition to that, lower reported incidence of pruritus and cholangitis adverse events. So with that, I would like to conclude by saying thank you. Participation in clinical trials, well, I'd love you to participate in Pliant's clinical trials. I think participation in clinical trials in general is really, really important. And every step you take, everybody who participates in a clinical trial is helping to push that ball forward. So thank you all very much, and have a great rest of your meeting.
Thank you very much, all of you. That was great. And I'm thrilled. At the end of the day, we've had this long day, and you can see everybody is still here. So thank you all. You're a wonderful crowd. So originally, we were going to have a short Q&A, but I know everybody's tired, and so we are going to let you out a little bit earlier so you can relax and you'll be in a good mood for dinner. But I would just like to say, I hope, so if you have any questions for any of these pharmaceuticals, please stop by their booth. They'll be all day there, so please think about it. And I hope you will consider participating in a trial, because the more of us that can do that, the faster we're going to find new therapies and eventually a cure. And if you want to see more information, learn more, if you go onto our registry site on the website, we'll find all the updated information about clinical trials. And the last thing I just want to say is that I hope you all get a message, but if you didn't see it, so because of the weather, which is not very nice tonight, a lot of wind and I guess sand is blowing, we don't think that would be fun to be outside. So we're going to be in the Canyon Ballroom, and I know it's hard to find everything, but Audra has just put up a new map on Cvent that tells you how to get to it. So basically, it's just across the street of this center. And if you went to Enchiladas yesterday, it's right on the way there. So it's across the street from the center. It kind of looks like a gymnasium almost. So it's a long building. I hope you can find your way. And Joanne, will we have volunteers to help them get there? Yeah. So watch for volunteers, okay? Thank you very much. I will see you all at 6:45, okay?